Good morning, everyone, and welcome to our session number 12 of the Research Data Management Workshop series. We would like to start acknowledging that we pay respect to Algonquin people who are the traditional guardians of this land. We acknowledge their long-standing relationship with this territory, which remains unceded. We pay respect to all Indigenous people in this region from all nations across Canada who call Ottawa home. We acknowledge that traditional knowledge keepers, both young and old, and we honor their courageous leaders, past, present, and future. So just a few announcements. We'll have our next two and last sessions of the Research Data Management Workshop on December 5th and December 12th. With Dr. Jennifer Miller from Yale University talking about data sharing, research equity, and the role of Good Pharma Scorecard, and with Kate Spector-Baghdadi on December 12th with patient preferences for data sharing and privacy considerations, intersection of law and health, and she's from the University of Michigan. So the registration will be open by the end of the day, and you can find all the information on our website. So we would like also to invite you to be part of our project. So if you are interested in research data management and data sharing or open science, we invite you to join us. You can find all the information on our website, and you can learn more about our next initiatives and how you can join the team. So today we have the session toward an open science ecosystem in neuroimaging with Dr. Russel Poldrack from Stanford University. Well, the session usually takes around 40 minutes, and we have 20 minutes for questions and answers. So we encourage everyone to place the questions in the chat, or after the presentation, you can also open the mic and ask him directly. Thank you so much, Dr. Russel, for being here with us. It's a pleasure. And I will stop sharing my screen so you can take the stage.
All right. Well, thanks very much for the invitation. Nice to meet you all. Yeah, if you have any questions as we're going, feel free to either speak up or type them in the chat, and I'll be watching that. Cool. So I want to talk about basically how open science and data sharing have emerged in the context of the particular research domain that I work in, which is brain imaging. And let's see. So just first, for my conflicts, I have research grants from the NIH, and I'm a paid consultant for NIMH. So I think we probably all agree, if you're here for this talk, that transparency is one of the essential components of reproducibility. This is just one of the many ways to kind of chop up the domain of reproducibility from the Turing way. And so what they refer to as reproducible is sort of same data, same analysis. So that seems like that's the kind of the minimal sort of reproducibility that we could ever hope for. That if given the same analysis of the same data, you can get the same answer. And it's pretty clear that in order to do that, you need to be able to access the data. Another framing of this I really like comes from this paper from a while ago by David Donahoe and Buckheit, where they were kind of talking about the work of John Claerbout, who had been an early advocate of open science in the computer science world. And they said, an article about computational science in a scientific publication is not the scholarship itself. It's merely advertising of the scholarship. The actual scholarship is the complete software development environment and the complete set of instructions which generated the figures, which for us would also include the data. So in some ways, the field that I work in, brain imaging, is a kind of a best case scenario for open science and data sharing. And this is focusing primarily on research using magnetic resonance imaging or MRI to study human brain structure and function. And, why is it a best case scenario? Well, one, the data are digital pretty much end to end. We collect them on an MRI scanner. They come off that scanner in digital format. We do automated analyses. There's usually very few or no manual analysis steps. There's quality control usually that's often manual, but in terms of the analysis steps themselves, it's usually not, based on any kind of manual process. We've also largely converged, one, on a standardized image format that's an open format, and a relatively common spatial coordinate system for describing where in the brain activity sits. And I'll show you a bit about that later. Now, interestingly, our field actually had sort of a false start back around 2000. A group at Dartmouth started this project called the fMRI Data Center. And it was started by somebody who's a very big name in our field, and who was an editor at one of the journals and a member of the National Academy, who actually pushed a couple of journals to start requiring researchers to share their data on this platform prior to publication. And this led to a bit of an outroar. There were some, editorials in Nature Neuroscience and Nature. And this was a text on the right from an open letter from a group of scientists who were saying, they're particularly concerned with any journal's decision to require all authors of all fMRI related papers accepted for publication to submit all of the experimental data to the data center. So clearly the field wasn't ready 20 years ago for data sharing. I think one big thing that changed, was in 2010, where we saw sort of a landmark paper. I think this, you really can say that this is the year that data sharing finally broke in neuroimaging. And this was a paper from a large group of investigators across the world who shared data that each of them had collected with one another to put together a sample of over 1,400 individuals, which was, an order of magnitude bigger than any of the samples that had been published prior to that, and used it to really show some interesting novel scientific findings about brain organization that had not really been evident from previous work with smaller samples. I think it's fair to say now that data sharing is becoming or has become the norm in neuroimaging. And I'm going to talk throughout the talk about various different types of data sharing, and those are kind of laid out in the framework that I show here on the left from our annual reviews paper a couple of years ago. We can also see on the right from a figure from Mike Millam's paper a few years ago, where he basically, laid out the number of publications that have been based on several shared data sets from a group that he runs called the International Neuroimaging Data Sharing Initiative, or INDI. A couple of years ago, an anonymous senior researcher at the Organization for Human Brain Mapping meeting, which is one of the main meetings for neuroimaging researchers that my group attends, was heard saying that OHBN has been taken over by the open science zealots. And that person has now become an open science zealot themselves. So that, I think, tells you just how far our field has come. So I'm going to talk today about 3 different projects that I've been involved in, and then sort of some of the infrastructure that supports them, that together comprise a sort of an open ecosystem for retrospective data sharing. There's a number of projects that have been going on focused on prospective data sharing, that is the collection of data sets meant primarily for the sharing of those data. The projects that I'm going to talk about are basically meant for sharing data that have already been published by researchers. And they vary in the kind of data that they share, with Neurosynth focusing more on breadth versus depth by sharing kind of published coordinates of neuroimaging data. On the other extreme, OpenNeuro, much narrower but much deeper in sharing kind of full raw and processed neuroimaging data sets. And I'll talk about kind of where these come from and how they work. One of the principles that we've really based all of our work on is sharing data as maximally openly as possible. And we think that in part this is important to honor the contributions of our research subjects. Any limits on data sharing are necessarily going to limit the benefits that can be gotten from the data. And as human subjects researchers, we've made a compact with our subjects to both minimize the risk and also maximize the benefits of their contribution. So all of our projects, NeuroVault and OpenNeuro, both use what's called a CC0, or a public domain dedication. And I have on the right here the definition of CC0 if you're not familiar with it. But this is basically as maximally open as one can share something, because it basically puts the data into the public domain. One of the benefits of this is it allows us to make all the data available programmatically via web APIs and other sort of programmatic systems, as well as via a web page. So Neurosynth is a database that shares published activation data from neuroimaging papers. So one thing that one commonly sees in a neuroimaging publication is something like this table with data showing where was there activity in the brain, where were the clusters of activity in the brain for some particular analysis. They're described in terms of their location in this XYZ space that you can kind of see up here. X is right, left, Y is front, back, Z is top, down. So those three numbers tell you where in that standard frame that activation was found. And it's not perfectly standardized, but it's fairly well standardized. So Tal Yarkoni developed a tool that he calls automated coordinate extraction that allowed us to basically grab these activation coordinates from papers automatically for at least for a number of different journals. And the current database has over 14,000 papers worth of data in it. Actually, I say full text, it's not full text, it's the abstract text. And what we do is we basically take those coordinates, place a little sphere at each of those coordinates, and we can think of that as kind of like the reconstituted whole brain map of activity that comes from that very sparse, small set of numbers. And this was validated against a manually curated database, and it does, fairly well. If you go to neurosynth.org and put in a search term like working memory, you'll see something like this, which is a meta-analytic map, basically an automated meta-analysis of all the studies that mention that term working memory in their abstract or title. And this, in this case, that comes up with about 1,000 studies, and it shows you the brain areas where that the presence of that phrase or working memory in the abstract is associated with activity in that particular part of the brain. And you can also download this map to do further analysis on it. This has become a really commonly used tool in the neuroimaging literature. Here's just one example. This is from a paper published in PNAS a few years ago by Dan Margulies and colleagues. They've come up with a new way of analyzing what they call principal gradients across the brain, which are basically kind of gradients of functional organization. And so what they did was they took the each of those particular gradients and put it and basically ran it against the Neurosynth database to do sort of an automated meta-analysis to ask for each particular gradient, which topics is it most associated with? And this shows, for each of them, some of them are more associated with things like social cognition or motion, some with more sensory motor things like motor, pain, eye movements, and so on. And so basically, this just shows how this database has enabled people to do automated meta-analysis on brain imaging data to provide sort of functional annotation of any particular brain image. So, beyond, so the database that I just showed you, Neurosynth, is containing, highly sort of, sparsified representations of any particular map. But the actual data that gave rise to those tables are usually on the order of about 200,000 statistical values that we threshold and then cluster. And often we'd like to have our hands on the actual whole brain map rather than the coordinates. And so what NeuroVault was a tool developed by Chris Gorgolewski and my group to basically allow researchers to share whole brain statistical maps that are unthresholded. And so if you go to the NeuroVault website and just look at one of the data sets, you'll see something like this. Any particular data set is composed as a collection of images. So for example, you might have 10 different analyses that are presented for any particular paper. And that could be, each of those would be represented as one of the particular images in the data set. And so any researcher could come and upload their data as long as they're in the standardized format. One of the things that they then get is a persistent identifier that they can place into their paper, and then anybody can come and sort of find that from the paper itself. We also link back to the paper when an identifier is available. If you go into one of these individual images, you can poke around in the image to see the values. You can download the image. And then there's also a number of different analysis tools that we provide for researchers in order to be able to gain added value. One is to actually take this map and do exactly one of those kind of automated meta-analyses that I just showed you. We call here cognitive decoding. There's also other types of decoding or power analysis that one can run. But basically, this is meant to provide researchers with added value beyond just giving them a persistent identifier, which is, I think, the main reason that people usually come to the site. Here's just one example of a recent usage of NeuroVault. And this is work from Bertrand Thirionn's group in Paris, where they took NeuroVault data to basically generate a novel way to decode cognitive functions from brain activities. The idea is, given some particular map, I'd like to know, what is that person doing? Are they looking at a human face? Are they moving their right hand or their left hand, or so on? And so what they did was they took the NeuroVault maps and they combined it with an ontology that we've developed called the Cognitive Atlas, and basically used that to generate a decoder. And here on the right, they show that this decoder works substantially better than others like the NeuroSynth decoder at actually decoding particular cognitive functions from a brain imaging data set. Again, this is just one of the many examples of people who've reused NeuroVault data to do novel, interesting scientific stuff. So I'm going to focus most of the talk on the OpenNeuro database. This is at openneuro.org, and it's basically an open database for the sharing of a number of different types of neuroscience data. So I'm going to tell you a little bit about it. And first, I'm going to highlight the fact that, especially when it comes to sharing complete raw data sets, simply sharing the data is not sufficient, right? If I just, put the data up on an OSF link and don't really do anything, to organize it well, it's not going to make it very useful. And many of you, I'm sure, have had this experience of grabbing openly shared data from a repository and finding that you can't do anything with it without emailing the owner to figure out, what on earth the data actually is composed of. Data need to be shared in a way that make them useful. And I think this, if you haven't seen this YouTube video, data sharing and management snafu in 3 short acts, it's pretty humorous. This is a little snippet from the discussion on the right, which highlights some of the problems that many people have had with data sharing. So the sad panda is trying to get some data from another panda, and they say, I received the data, but when I opened it up, it was in hexadecimal. The other panda says, yes, that's right. I can't read hexadecimal. You asked for my data and I gave it to you. I've done what you asked, right? That's the attitude that many people in the past have had about data sharing. Is there a guide to the data anywhere? Yes, of course. It's the article that's published in Science. Clearly, this is not effective data sharing. So we decided early on, OpenNeuro grew out of an earlier effort that we had run called OpenfMRI that had used its own kind of bespoke format for organizing the data. And we decided that if we were going to sort of, make data sharing work more broadly, that we needed to have a community-driven standard for organizing the data. And so we worked with a number of researchers across the field to develop this thing called the brain imaging data structure, which has now become a very commonly used and pretty successful standard for organizing the file structure and the metadata of a neuroimaging data set. So this started back in 2015, where we held an initial meeting at Stanford and started working on a draft standard that was released later that year, and basically solicited feedback from the community. In addition to providing the standard, we gave a large number, 22 example data sets that people could look at to basically see, what a data set in BIDS looks like. In 2016, we published the paper in Scientific Data. And then a couple of years later, we really turned it into a community organization, moving it out of our labs organization into the BIDS standard GitHub organization, which it is now where it lives. So BIDS is really built around a number of principles that were in part informed by, what we had seen in the past as sort of, some failures of data sharing. The first principle is that the most important thing is getting people to use the standard. And we wanted to, in principle, in particular, keep it as similar as possible to what people are already doing in their own labs. And that often means not using the most technically sophisticated tools that one might use, because we didn't want to let technology override usability. We also wanted to focus on engaging the community to try to bring in as many people as possible and incentivize them to use the standard. A second principle is don't reinvent the wheel. Whenever there's an existing standard, we want to reuse that standard. And finally, we kind of pretty strongly adhere to what's often called the 80-20 rule, right? The idea that most of the effort in any particular project is going to go into edge cases. So you want to focus on the most common use cases and not let the perfect be the enemy of the good. Not let people who are griping about the fact that you can't address some particular edge case kind of derail you from doing work that's going to help most people most of the time. So when we get data off of an MRI scanner, it looks like what you see here on the left, and it's in a format called DICOM. And it's really not very useful in the way that it's organized in terms of how researchers think about data. On the right, you see those data reorganized into the BIDS standard. And if you're familiar at all with neuroimaging, these different labels will make sense to you. If not, basically you see that there's a separate folder for each of the subjects in the study. There's a different folder inside each of those for different types of MRI data. There's a standardized file naming scheme for naming the files. Each file has what we call a sidecar file that's a JSON file that contains additional metadata about that imaging file. And so this basically looks like the way that most researchers were organizing data in their labs, before BIDS came along. It just sort of, kind of cleans it up a bit. And one of the most important things that we set out to do when we started building BIDS was to be able to basically automatically validate a data set. We want to know, before a data set ever gets uploaded to OpenNeuro, is it actually a valid BIDS data set? And so we built this JavaScript BIDS validator that can basically take a data set and look at it, without ever uploading it and tell you, is it a valid BIDS data set? And if not, it can give you some feedback about what needs to be fixed in order to make it a BIDS data set. Now, one of the most important things I think about that this led to the success of BIDS was building in the ability to extend it to new types of data. And so we added in an extension process through what we call BIDS extension proposals, or BEPs, that was really patterned after the Python enhancement proposal, or PEP process, that's used to sort of add things in the Python language. And so you can see there are to date 11 different BEPs that have been accepted or completed. And this basically requires sort of, a number of researchers in the community to sign on to it. And one of the things this has done is allow us to extend BIDS into new types of data sets, like magnetoencephalography, PET, electroencephalography, and so on. Now, BIDS has grown substantially in its usage over the last 6 years. And one of the ways that we can see that is by looking at the data that have been uploaded to a system that we run called the MRIQC Web API. So MRIQC is a software tool that can basically take a BIDS data set and provide a quality control report about that data set. Quality control for MRI has often been sort of challenging, and MRIQC makes it really easy as long as your data are in BIDS. And so it's one of the incentives to try to get people to move their data into BIDS. One of the things that MRIQC does is whenever it's run on a data set, it uploads some summary statistics about the quality control to a web database, unless the user opts out of that upload. This telemetry system makes it really easy for us to see how often it's being used, and if it starts crashing, we'll see that. The data from this API are actually publicly available. We have a scientific data report from a couple of years ago that sort of laid it out. But what we see is, as of earlier this year, there were data from about 375,000 unique functional MRI scans and about 280,000 unique anatomical scans. These are all data that had to be in BIDS in order to be able to be run like that. So that's showing us that there's at least well over 250,000 data sets that have been converted into the BIDS standard at this point. Now, one of the things about BIDS is that it's really enabled the growth of an entire open source software ecosystem. So there's a ton of tools now that are basically built around BIDs and sort of related tools that kind of work together with BIDs that have allowed almost any type of data to be processed using open source software. And we think that this is really important to make, again, make things as open and transparent as possible to other researchers, to allow them to really reproduce not just our data, but our workflows. So let's go back and say a bit more about OpenNeuro. OpenNeuro is one of the official archives for the BRAIN Initiative, which is a cross-institute initiative within the U.S. that supports lots of neuroscience research. And basically, OpenNeuro supports the sharing of any validated BIDS data set. So whenever there's a new data type that's added to BIDS, we can, by just updating the validator, we can automatically add that as a type of data that can be shared by OpenNeuro. So if you go to OpenNeuro, you can actually look through all the data sets you don't need to sign in or anything. The data are all available to anybody for download. And this would be an example. If you go search for reading, you'd see several data sets about this particular project. You can see there's 194 data sets that match the search for reading. And there's some sort of information about each of the data sets. If we go to the page for one of those data sets, what you see is some information, kind of a README file about the data set. You actually see you can access the individual files. That we have a file browser that allows you to either individually download or visualize those individual files. Each data set is associated, and each version of each data set is associated with a persistent identifier, a DOI, such that when the researcher goes to publish the data, they can point to exactly the version of the data that they use. And these versions, I'll show you an example later of how they're created, but the versions are immutable. So once a version is created, that data set can't change. And that provides a highly reproducible version of the data used to create the analysis. So as I mentioned, any valid BIDS data set can be shared via OpenNeuro. So I'm going to kind of walk you through what the workflow looks like. So if you go to OpenNeuro, you see there's a sign in button up here at the top right. And currently we allow signing in either via Google credentials or via ORCID. Right now we don't do a great job of linking data sets up to ORCIDs. That's one of the big things we want to do in the future, is having a linkage between each researcher's ORCID and all the data sets that they're associated with. Once you log in, you see that now you have my account up here, and you can actually go, you could go look at the data sets that you own. I want to show you what happens when you click the upload button. The first thing you're asked to do is to select the data. And you'll notice we have a note here, and I'll come back and talk about this later. To protect the privacy of individuals who've been scanned, we require that all scanned data be defaced before publishing a data set. So the expectation is, at least under US law or US federal regulations, data that have been de-identified are not considered to be human subjects data. So they can be shared without any sort of special permission from the individual. And so that requires not just removing all of the usual identifiers like name and birth date and things like that, but any other information that could in principle be used to identify the person. And for MRI images, there's information about the face that could in principle be used to reconstruct an image of that person's face. And so we require that information be removed. Again, I'll have more to say about this later. So select a BIDS data set to upload, and then basically that gets uploaded. We also have other ways to upload data. Sometimes, if it's a huge data set, uploading it via the web is not always the most effective way to do that. And so we have a command line tool that you can also use to upload very large data sets. But here's what happens. So I uploaded a data set. and it says, we found it runs the BIDS validator. It says, we found 3 warnings in your data set. You're not required to fix them, but doing so will make your data set more BIDS compliant. There's a number of things that aren't required, but they're sort of suggested. And if those are found, so for example, tabular file contains custom columns not described in a data dictionary, you get a warning, but you can still upload the data set if you want to. So let's say that we go ahead and continue. Now we're asked to enter a number of bits of metadata about the particular data set. These metadata are not required, but they're encouraged for the researcher to include them. And this is using a metadata entry tool developed by Mark Muson's group at Stanford. So finally, you've entered your metadata, and now you have to accept the terms of sharing. The first is that you're the owner of the data set and you have the necessary ethics permissions to share the data publicly, and that you have de-identified them according to HIPAA, which is one of the regulations that governs the de-identification in the US. I agree that this data set will become publicly available under a Creative Commons license. We give people a grace period of 36 months. So you can initially upload the data and have them stored privately, and you can actually share them with individuals if you have collaborators you want to share them with. But the understanding is after 36 months, they're going to become publicly available. You have to agree that the data set is not subject to GDPR. We don't have the ability right now to share data in a way that would be compliant with GDPR. We're working with researchers in Denmark to try to develop sort of a new version of OpenNeuro that would allow sharing of the sort that is supported by GDPR. But right now, the data have to be openly shareable. And then you have to say either that all the scans have been defaced, or that you have explicit participant consent to publish their scans without defacing. Sometimes, for example, I have a data set that I've shared from myself that I don't feel the need to deface because everybody knows it's my data set. And so I would click that, I have permission to share those. Once you go about sharing, you are taken to a page where you see this is a draft data set that you haven't created any actual, shareable versions of it yet. And then once you're ready to create a new version, you would basically just, add some information about what you've done and hit create version. It'll create a new version for you. And then that version would get a DOI. And then you would see something like this, where it would show you, hey, here's the data set. And you can go about downloading the data set. And if you click on download, you'll see there's a number of different ways the data set can be downloaded, including via just downloading straight from the web, but also through more programmatic tools. So OpenNeuro has grown kind of, substantially and pretty, consistently over the last 5 years. And so here you see its growth from mid-2018 up to last month, I think. And you can see we're up now above 700 data sets, up above about 30,000 individual subjects. Most of those are human subjects. So this is on the right, you see a table showing the number of studies with each different species. So you see most studies are human studies, but we have, about 20 studies that have used mouse, 12 rat, 2 non-human primate, and a number of other ones. Most of them are MRI, including both anatomical and functional MRI. But you see, we also have other data types. We have, over 100 EEG data sets, 30 MEG, magnetoencephalography, 17 intracranial EEG. That's sort of, recordings from inside a human's head when they're undergoing surgery. We also, one of the things that we particularly like about this particular database is the diversity of the types of data sets that we see in terms of their kind of size versus breadth. And so this is a plot on the left of, on the x-axis, the number of participants in the data set. For humans, the number of people in the data set. On the y-axis is the number of sessions, how many how many runs have been of data, or how many different scans have been collected for each individual, which is usually one, but it goes up to over 100. And then the size of the data point shows you the number of tasks, which is the number of like different things that are being measured about the person. Usually it's a small number, but we have some data sets that are up to say 48. So it really shows that there's a very broad diversity of data types in the archive. The data sets have started to be reused pretty substantially. This is from the paper that we published last year. Shows through mid 2021 the number of reuses. The 2021 bar is only through the first half of 2021. Unfortunately, there's not an easy way to generate these publication counts. It really requires kind of manual searching. So we haven't updated this particular figure into this year, but we're pretty sure that it's continued to grow. But nonetheless, you see that there's a decent number of both journal papers and preprints that have reused data from OpenNeuro. Here is one example of such a reuse that comes from work that we just presented this week at the NeurIPS meeting. And it really tries to address a challenge for functional MRI studies, which is that most of the data sets are very small. And when we try to apply machine learning tools to these data sets, machine learning tools are very data hungry, right? They usually require much larger data sets than we have in order to train them properly. And so Arvin Thomas in my group had the idea to create what we call a foundation model, kind of based on work that's been ongoing in the development of large language models in AI. Basically, we took a large amount of data from OpenNeuro and created a pre-trained model by just training the model to learn the structure of those data that we could then use to go and perform decoding on an independent data set. And we see that that this approach of pre-training a model that's then applied to a new data set, rather than training a model de novo on that same data set, performs much better at decoding, what a particular person is doing at any particular point in time during that study, compared to kind of the standard baseline models. This is just one example of how we can take advantage of the size of OpenNeuro to generate novel scientific findings. Now, I mentioned that all OpenNeuro MRI data sets must be defaced to reduce the risk of de-identification, but there's ongoing work that's been looking at whether one could, in theory, take these defaced data and still effectively apply face recognition systems. So, particularly Christopher Schwartz and colleagues have published a couple of papers that have suggested that one can take defaced data, impute a face based on a model, and then apply facial recognition tools, and really taking advantage of the fact that there's structure, in the shape of the head, the shape of the brow, that aren't being removed by defacing, that one can, with, non-zero accuracy, re-identify an individual from their defaced brain image. And obviously, if that risk continues to rise, we may have to move away from the type of open sharing that we've been engaged in. And we think that would be a huge loss for researchers, and for research participants, right? Because it would limit the benefit of shared data substantially. So we've recently proposed regulatory changes that are, in the US context in particular, to try to protect subjects from misuse of neuroscience information, kind of patterned after a regulation that's been on the books here for a while called the Genetic Information Non-Discrimination Act, or GINA, which basically prohibits employers or insurance companies in the US from using genetic information to make any sort of decisions. And we proposed in this paper by Annie Joie in my group that we need an analog we call the Neuroscience Information Non-Discrimination Act. And the hope is that, if we have regulatory improvements, in addition to potential technical improvements of defacing, that we may be able to, retain the ability to share data openly while still protecting subjects' confidentiality, or protecting them from the risk of re-identification. So why has neuroimaging data sharing been so successful? Well, I've already mentioned, one key to success, which is the fact that the data are digital end to end, right? It makes it very easy to share. And the fact that we have standardized file formats like the nifty format that I mentioned, and data standards like the BID standard. And there's an increasing prevalence of tools that allow researchers to fairly easily get their data into the BID standard. So we reduce the effort for the individual to curate their data, and then we also reduce the effort on the data sharing archive for data curation, which helps those archives continue to run in the long term because running an archive can be expensive if you have to hire curators. I think most importantly, in our field, there were very early demonstrations and continued demonstrations of scientific utility of shared data set, and a lot of success stories of researchers who have basically built careers out of using shared data. So what are the lessons that we've learned? I think one is that, getting the community to buy in to data sharing is essential. We already saw the example of the fMRI data center where trying to put mandates in place too early backfired. Unless there's, we saw in genomics, you had very powerful advocates that basically made data sharing happen. If you don't have that, then basically you need the community to buy in. And it's, in Silicon Valley, there's a term that people use called dog fooding, which basically means using your own product. And so I think one other thing that's been important is that the people who've been advocating data sharing, like myself, like Mike Millam, like a number of others, aren't people coming in from the outside saying, hey, you need to share data. We've been researchers from within the field who basically come and said, hey, we need to move towards sharing data, and have really sort of, walked the walk, ate our own dog food and showed, hey, we're going to share our data publicly. I think another lesson we've learned is to sort of keep it simple and keep it as close to what people are already doing as possible. I'm showing you on the right an example of an early data sharing project. This is their informatics architecture for this project called BIRN, Biomedical Informatics Research Network, which was trying to share data and basically built such an over-engineered system that no one knew how to use it. And it basically went nowhere. And it was a very, technically sophisticated system, but it was just over-engineered. And, I have a kind of a rule of thumb, which is if there's more than two novel acronyms, then you might want to be worried about whether your tool is too complicated for people. I think more than anything else, kind of adhering to the Pareto principle has been really important for us. Not letting the perfect be the enemy of the good, focusing on the 20% of the effort that'll cover 80% of the data sets, and realizing that there's always going to be a long tail of edge cases. Those edge cases are going to have loud advocates. And so you're always going to have some people saying that your standard is useless, your tool is useless because it can't, accommodate their particular edge case. And you have to look at those edge cases and make sure that you're not missing out on something important that could be incorporated, but then realize that, making something that works for most people most of the time is in the end going to be of substantial benefit to people in the field. So in conclusion, I think our field has built a model ecosystem in a lot of ways for open science and data sharing. And we've really found that building usable infrastructure is critical to easing the friction that people often experience in data sharing. And really engaging the community and convincing them of the utility of open science and data sharing has been really important. And realizing that, when we build these kind of tools, we need to keep them as close as possible to what researchers are already doing in their lab. So with that, I'll end and thank our team, our collaborators, and the folks who've funded our research. And thank you.
Thank you so much, Russ, for your excellent presentation. So first, I would like to ask if anyone has a question to Russ. I cannot see anything here in the chat. So if I can start, you mentioned a lot about the community engagement and how this will be the key for improving data sharing. How do you see the role of institutions and funders in place in terms not only to increase and to encourage data sharing, but also in terms of harmonization of models and standards? How do you see that this should be somehow implemented?
Yeah. I mean, I think that for me, it all, a lot of it comes down to incentives, right? So at the institutional level, I think that the most important thing that we can do is to provide researchers with really strong incentives to do, the kind of work that many of them, I think, want to do, right? Which is open and transparent work. And, right now, in most institutions, people don't necessarily receive credit in the context of tenure and promotion or hiring, for doing open science, right? The credit model still focuses on high impact publications, very clean stories, not necessarily, being open and sharing. And so for example, there's an effort ongoing in the US right now called Helios, Higher Education Leadership Initiative on Open Scholarship, which is really trying to move the needle on getting a large number of institutions together to come up with models of how we can better incentivize open scholarship, data sharing, open science, in our case, in the context of hiring and promotion and tenure. I think that on the funder side, the one thing that obviously they have a very strong handle on being able to, mandate sharing, and that's certainly happening in the US now. The National Institutes of Health, that as of a couple months from now, is going to require mandatory data sharing, data management plans, and ultimate sharing of the data. And I think that's a really important move. And also the idea that that the costs of data sharing are something that should be funded within the grant, right? Often it's been the case that you might want people to share data, but they can't, they're not allowed to ask for money to actually cover the costs of doing, there's extra work to share data, right? Getting your data into the bid standard and so on, in our case, takes time and effort. And so, researchers need to have those expenses covered. So I think that those are the really important things on the part of institutions and funders. I mean, I think in terms of harmonizing efforts, I think it's hard to expect institutions and funders to really be at the forefront of that because it's so field specific, right? It's so specific to the type of data that you're sharing. And it's really the role, I think, of the professional organizations and the professional societies to be at the forefront there. So for example, the society that I'm most involved in, the Organization for Human Brain Mapping, has for a number of years been involved in, has had a community on best practices. We published some papers laying out best practices in the field around sharing, motivated using bids, and so on. And so I think figuring out a way to align the efforts of the professional societies with with what's going on in the institutions and in the funders, that's probably the best way to to kind of bring everything together.
Thank you, Russ. We have a question here from Rebecca Thornhill. She said, fantastic talk, thank you. I'm curious about attribution and acknowledgement of the source of open data in the literature. To date, how does your particular research community feel about this? Do you feel your work or shared data are properly acknowledged in manuscripts and etc.? Thank you.
Yeah, I think that's a really great question. It sort of depends on what you mean by properly acknowledged. I think in general, when people reuse data, they do provide, I think, almost everybody will provide some sort of citation to the data set that they're using. And so I don't think we have a lot of cases of people taking data and trying to publish them, without acknowledging that they were shared. And in part, that's just because in our field, it's become perfectly fine to write a paper about a shared data set, right? People aren't expected to necessarily, collect the data themselves. And when occasionally there have been cases, I think, where people tried publishing data without acknowledging the provenance, but for the most part, there's not a cost to acknowledging that, and so we haven't seen that as a problem in our field. Now, properly, if by properly you mean in a way that can actually be found straightforwardly, the answer is no, right? That what we'd like is anytime anybody reuses the data set, they will include the DOI of the data set that they used. And people are increasingly doing that. I think there's a growing realization of the utility of persistent identifiers. But, I mentioned, coming up with that figure in our paper of the number of reuses, we had to have a research assistant go in by hand and search in data sets. And often, sometimes, people would just, instead of putting a DOI, they would just say, we reused DS blah, blah, blah from OpenNeuro. Without providing a link to, or even using the recommended citation language, that we provide, that the owner of the data set provides in their data set. So there is, like, DataCite has this tool, like if you use the DOI, then you can sort of link back to the data set. But it's still not as good as we would hope.
Thank you. Rebecca also mentioned that this was exactly what she was wondering. Do we have any questions? Please, you are also welcome to open the mic and ask directly to Dr. Russel. We still don't have. So you mentioned about the White House policy that is coming about open access and open data. I think there was a fuzz in terms of open access, the aspect of open access, but not necessarily of the availability of open data that is also there in the policy. Right. And this, I don't know why, but it catch less attention than the part of open access. How do you see this policy incentivizing? I think it could be a shift of open data, certainly, and how to encourage and improve open science.
Right. I think that, to some degree, the open data language that was in that Nelson memo, in our world, if you're, in terms of like people who get funding from the NIH or the NSF within the US, they're already on top of this stuff. So I think it's not going to be sort of such a big, that particular memo didn't have a big impact, simply because, NSF has required data sharing for a little while. NIH is going to be, NIMH has required it for a little while already. The entire NIH is going to be requiring it as of this coming year. And so I think they were already ahead of the curve in terms of kind of requiring, data sharing. I know there's some other agencies that I think are going to be coming along, in the wake of that memo that weren't requiring data sharing that are going to be. But I think in terms of like biomedical and basic science research, in the US, those agencies were kind of already on board. And so I think that's why, a lot of the focus was on the open access language, because that is, and again, NIH has already required open access through PubMed Central for a while, but I think that this is gonna help accelerate the move, towards open access in general.
So I don't see any questions here, so I can give back to everyone this last minutes. Thank you very much, Dr. Russel, for being here with us. It was a great talk, and all right. See you all very soon for the next session.
Great, thanks.
Have a good day.